Fundamental unit of life. Topic: Transportation across membranes. Students, in this topic, I'll be telling you about passive transportation, under which we'll be discussing about diffusion and osmosis. Then I'll be discussing about active transportation and then about bulk transportation, where we'll be learning about how in bulk materials are taken into the cell by the process of endosmosis. To start with, let us first have a closer look at the structure of plasma membrane. Now, as you all know that every cell is covered by an outermost thin membrane called as a plasma membrane, which I've already told you it's thin, delicate and highly flexible. Now this plasma membrane is made up of lipids and proteins. This is the structure of plasma, the structure that you see here. This is actually the structure of plasma membrane. Now, as already told to you, that plasma membrane is made up of lipids and proteins. So here, let us see how these lipids and proteins are arranged. What you see as these blue structures, these are actually the lipids. So lipids in the plasma membrane is arranged in the form of two layers. There's a two layer of lipid present, and apart from these two layers or bilayer of lipid, you also find that the plasma membrane or this bilayer has got proteins embedded in it. So these two are the important components of plasma membrane. Apart from this, you also find there are some carbohydrates also attached to this bilayer. Okay, now one more very important thing to tell you is that this membrane is not just present outside the cell, but then the organelles, about which you have learned in class 8 also, that every cell has got organelles, right? So like mitochondria, there's endoplasmic reticulum. A detailed structure of all these organelles will be dealt later on, but these organelles are also bounded by membranes, okay, which separates the inner content of these organelles from the outer cytoplasm. So these membranes also have the same structure. Membranes can either be selectively permeable or semi-permeable. A selectively permeable membrane is a membrane which allows solvent and some selected solutes to cross through. You have learned that plasma membrane is a selectively permeable membrane. Why we call this as a selectively permeable membrane? Because this plasma membrane allows selected substances to move in and out. This was already dealt in my previous video, right? So if the membrane is allowing solvent and some selected solutes to move in and out, like if suppose if you look at this diagram, then if this is the outside of the cell and this is the inside of the cell, then the membrane, if it's allowing solvent to move in as well as some selected solutes to move in or out. Okay, so the membrane which allows solvent to move in and out and some selected solutes to move in and out, such a membrane will be called as a selectively permeable membrane. Membranes can also be semi-permeable. A semi-permeable membrane is a membrane which allows the passage of only solvent. So if a membrane allows only solvent to pass through but does not allow solute to move in and out, then that such a kind of membrane will be called as a semi-permeable membrane. Now this plasma membrane behaves like both a selectively permeable membrane. At times it behaves like a selectively permeable membrane, and at times it behaves like a semi-permeable membrane. Now let us study in detail about how this transportation occurs across the plasma membrane. Transportation across plasma membrane can be either passive or it can be active. First, let us discuss about passive transportation. So passive transportation is movement of molecules. Have a look at this definition. Now, passive transportation is the movement of molecules from the region of their higher concentration to the region of their lower concentration without expenditure of energy. What does it mean? If molecules, if substances, like have a look at this structure which is shown over here. In this structure, what is shown that if suppose molecules are moving from the region of their higher concentration, if this is the outside of one side of the plasma membrane and this is the other side, then if the molecules move from the region of their higher concentration, here the molecules you find is in higher concentration and it towards the other side it is a lower concentration. So when molecules are moving from the region of higher to lower concentration, in this case the cell does not spend any energy, and as the cell does not spend energy, then that type of transportation is called as passive transportation. There's another type of transportation that also occurs across the membrane, and that is called as active transportation. Active transportation is the movement of molecules of solid, liquid or gases from the region of their lower concentration to the region of their higher concentration. So active transportation is actually the reverse of passive transportation. In passive transportation, molecules move from the region of higher to the region of lower concentration, whereas in active transportation, the molecules have to move against the concentration gradient, that is from the region of lower concentration of the molecules to the region of higher concentration. And whenever this kind of transportation occurs, in that case the cell has to spend energy. Okay, so in case of active transportation, the cell, this movement will occur with expenditure of energy. And some of the examples of active transportation as which happens in the cell is glucose, amino acids and many of the ions, they pass through the plasma membrane by this process of active transportation. When the root hairs absorb water, that is by osmosis, about which you'll be learning, but when minerals are absorbed from the soil by the root hairs, it occurs by the process of active transportation. Let's now study about passive transportation. Passive transportation, as already just now I've told you, it is a movement of molecules from the region of higher concentration of those molecules to the region of lower concentration without expenditure of energy. Now passive transportation is of two types, that is diffusion and osmosis. Let us learn about these two processes in detail. Now let's see what is diffusion. Diffusion is the movement of molecules of solid, liquid and gases from the region of their higher concentration to the region of their lower concentration. So diffusion, I'm repeating again, is a movement of molecules of solid, liquid and gases from the region of higher to the region of lower concentration. A simple activity that can be performed to show diffusion is, like suppose you take a conical flask. This is a conical flask. In a conical flask, if you add some dye like ink or something, what you find is that the dye first will be present in a particular region. As you drop it, it will be present at a particular region, and slowly this dye starts moving to all sides. That is, it starts moving from where? From the region of its higher concentration to the region where it is not present, to the region of its lower concentration, and at the end you find that the entire solution will be uniformly colored. So this activity shows the process of diffusion. Once again, let us see. Now here what you find is when the dye is placed or put in water, then the molecules of the dye, you see the molecules of the dye from the region of their higher concentration slowly move to the region of lower concentration, and finally they will go on moving and will be spreading throughout the solution till an equilibrium is attained. So till the equilibrium is attained, this happens. Remember that for diffusion to take place, a semi-permeable membrane is not required. So these are some day-to-day life activities in that we see diffusion taking place. Now let us study about how diffusion occurs, or what is the importance of diffusion in the living organisms or in the cell. The best example of diffusion inside the cell or taking place is the exchange of respiratory gases. Now, in a respiring cell, what do you find? When a cell, this is a diagrammatic representation of a respiring cell and how transportation of respiratory gases occur. Oxygen and carbon dioxide will move in and out of the plasma membrane, will move across the plasma membrane or in and out of the cell by the process of diffusion. So why and what you find is that always in a respiring cell, oxygen will enter the cell, whereas carbon dioxide will leave the cell, and this occurs by the process of diffusion. So why this happens, let us see. Now in a respiring cell, as a cell is respiring, you find that the inside the cell glucose is oxidized to produce carbon dioxide and water. So in the process, oxygen is continuously consumed inside the cell and carbon dioxide is continuously liberated. As a result, you find that the concentration of carbon dioxide inside the cell will be always high when compared to outside. If this is the outer part of outer medium of the cell, then the concentration of carbon dioxide inside the cell is always high because glucose is being continuously consumed or it is being continuously broken down and carbon dioxide is being produced. So when the concentration of carbon dioxide is compared from inside the cell to outside, you find that the concentration of carbon dioxide inside the cell is quite high when compared to that of outside. As a result, carbon dioxide diffuses from the region of higher concentration inside the cell to the region of lower concentration outside the cell. At the same time, let us see how oxygen diffuses. Now, oxygen is continuously being consumed inside the cell. Glucose is oxidized, and for oxidation, for respiration, oxygen is required, right? So, since oxygen is being continuously consumed, you find that the concentration of oxygen inside the cell is always lesser when compared to the concentration of oxygen outside the cell. As a result, oxygen from outside, that is from the outer medium, will move into the cell, right? So this diffusion of gases, that is oxygen from outside enters the cell in the respiring cell, and carbon dioxide moves out of the respiring cell, and this occurs by the process of diffusion. Now study about importance of diffusion in the living beings. Diffusion is required for intracellular distribution of substances. Intracellular means inside the cell, within the cell. Inside the cell you find that if this is a cell, then the different materials which are brought into the cell or which are produced inside the cell has to be distributed to all parts of the cell, and this distribution of materials within the cell occurs by the process of diffusion. Second importance of diffusion is that diffusion is required for exchange of respiratory gases. Just now I have told you how gaseous exchange takes place inside the across the plasma membrane. In a respiring cell, you always find that oxygen always diffuses into the cell, whereas carbon dioxide diffuses out, and this exchange of gases occurs by the process of diffusion. Next important function of diffusion is transpiration, that is the loss of water from the aerial parts of the plants. Okay, this process occurs by diffusion. Okay, so this is the, just have a look at the structure, this picture. Transpiration, it is a loss of water from the aerial parts through the stomata which are present in the leaves and through the other aerial parts water is continuously lost. This you have learned in your junior classes also. So this loss of water from the aerial parts of the plants occurs by the process of diffusion. Next importance of diffusion is osmosis, about which you'll be learning in the next topic, is also a type of diffusion that occurs through a semi-permeable membrane. Remember, I've told you a semi-permeable membrane is a membrane which allows solvent to pass through but does not permit the solutes to move across it. Study about osmosis, which is the movement of water molecules from the region of higher water concentration to the region of lower water concentration across a semi-permeable membrane, right? And this definition, if you just relate it with that of diffusion, what you find is osmosis is actually diffusion of water or solvent across a semi-permeable membrane. And try to recall, I've told you about what is a semi-permeable membrane. A semi-permeable membrane is a membrane which allows water or solvent to pass but prevents the movement of solute. It will not allow the solute to pass, but it allows water to pass. So here this is the diagrammatic representation showing osmosis taking place. If suppose a system is divided into two, like the system, it is divided into two by a semi-permeable membrane, and you fill both the sides of the system with solutions of different concentration. One side you put a dilute solution and the other side you put a concentrated solution. What you find is that when the two solutions are of different concentrations are separated by a semi-permeable membrane, you find after one side the level of this solution on one side will be increasing, and this increase in the level that you see is actually because of water moving from the region of higher water concentration. This side, what you see here, the free water molecules are more in number when compared to the solution which is concentrated. So water from the region of its higher concentration, here this side there is higher water concentration, so from here water moves to the region of lower water concentration. There is no movement of solute. The solute particles are not moving. It is only water passing through this semi-permeable membrane, and then water moves to the other side, and this is what is called as osmosis. Okay, so this is known as. Here is an activity to demonstrate osmosis. In this activity, we make a setup. So just have a look at this setup. This is the activity, thistle funnel activity, where we take a thistle funnel. This funnel that you see, this one is called as a thistle funnel, which has got opening at the base. The base will be, there will be an opening present. So what we do is we take a thistle funnel, and the bottom of the thistle funnel here, this side, we close it with a semi-permeable membrane. So parchment paper or animal bladder, these, or the cellophane sheet, these are all examples of semi-permeable membranes. So you take a semi-permeable membrane and put it at the base and fix it properly with a proper rubber band or something. You have to fix it properly so that there's no opening left from outside to things to enter in inside. Okay, so this is what is to be done. Now what you do is the from the top, that is from the top of this thistle funnel, from here, from the top of the thistle funnel, we have to pour sugar solution. Okay, you pour sugar solution, and then what you do is you mark the level of sugar solution. Now this thistle funnel with the sugar solution has to be placed in a beaker. You take a beaker, and in the beaker you place the thistle funnel, and this beaker has to be filled up with water. So you fill up the beaker with water, and now you leave the setup for some time. For some time you have to leave the setup. So when you leave the setup, so let's see what happens after some time. When you, so this is the initial part of the activity where you just mark the level of this water in the thistle funnel, and the funnel has got sugar solution, and exterior in the beaker there is water present. So leave it for some time. After some time, when you see, you find that the level of water in the thistle funnel after some time increases. There's an increase. Okay, now this increase is, and then you will also find there will be a decrease in the level of water. In the beaker, the water level also a little bit will decrease. Okay, now if you taste this water, if you taste this water which is present in the beaker, the water that is present in the beaker, if that is tasted, or you can test it also using Benedict reagents. So when you test this water which is present in the beaker, you will not get a positive test for sugar. This means that from the thistle funnel, the sugar has not gone into the water that is present in the beaker. But then the level of, here what you find is the level of water has increased, solution which was there in the thistle funnel has increased. So this shows that actually it's the water which has entered into the thistle funnel. So this activity shows that osmosis, it demonstrates osmosis. What happens here is that when you make the setup of this kind, where you have taken the thistle funnel with sugar solution, you find, and the outside of it you have placed a beaker with water. What you find, and the two solutions, that is two of the medium, that is sugar solution and water, how are they separated? They are separated by the semi-permeable membrane. The rise in the water level after some time is because water from the beaker actually enters the thistle funnel. And why? It's not that water is not going out, it is water from the beaker entering into the thistle funnel through the semi-permeable membrane. And this happens because the concentration or the amount of water, free water molecules in the beaker is much higher when compared to water in the thistle funnel. So this activity clearly demonstrates osmosis. There's another activity that you can do at home also. In this activity, what you do is you take a potato. Okay, so you take a potato like this, and then just peel out the outer peel of potato, and then base of it has to be made flat. Here the base of the potato has to be made flat, and then you make a cavity in the potato as shown over here. Right, now you do what you do is, the cavity of this potato has to be filled up with sugar solution. In this cavity you have to put sugar solution, and you just take a glass or a container in which you will place this potato, and in the container you have to take water. Now what you do is you mark the level of sugar solution which you have put in this cavity, right? And then you can take a pin and mark it, right? So after marking it, you just leave it for some time, say half an hour or so, you leave it. After half an hour, you'll find that the level of water or the solution in this cavity of the this potato will increase, and this increases because water which is present in the container that is present, that's in which the potato was placed. So water from here passes through the semi-permeable membranes of potato. Potato tuber is made up of lots of cells. So it crosses the semi-permeable membranes of the cells of potato, and finally this water enters into this cavity, and this occurs because here in the beaker the water concentration amount, number of free water molecules is much higher when compared, because inside you have taken sugar solution. So here you find that the free water molecules is less, and so water enters. Had you have been put, like if you would have put in the solution, in the cavity, if you would have put water and outside also there was water, then in that case there you will not find any change in the water level. Water will not move, because per unit volume, in that case, if suppose outside there is water and in the cavity also, in this cavity also there is water, then since the free water molecules per unit volume, per equal volume, if you take, will be the same, you won't find any change. Another thing is that if suppose instead of taking a fresh tuber of potato, if you boil the tuber of potato and then again do this activity, you will not find any increase. Reason is that when you boil the potato tuber, the cells of potato die, the membranes become ineffective, and so that is the membranes are dead, and so the entry of water in this cavity will not occur, osmosis will not be performed. So this activity demonstrates osmosis. Let's now differentiate between osmosis and diffusion. Diffusion is the movement of substances, molecules of solid, liquid and gases from the region of higher to the region of lower concentration, whereas osmosis is the movement of water. It is the movement of water from the region of its higher concentration to the region of lower water concentration across a semi-permeable membrane. For diffusion to occur, a semi-permeable membrane is not involved. There is no requirement of semi-permeable membrane, but osmosis will occur only when there is a semi-permeable membrane, right? Then diffusion occurs in solid, liquid and gaseous medium. It can take place in solids, liquids or gases, but osmosis always occur in liquid medium. Why? Because osmosis is the movement of water or solvent. So osmosis always occur in liquid medium. Now, osmosis is of two types: endosmosis and exosmosis. Endosmosis is the osmotic gain of water by the cell when it is placed in a hypotonic solution. Now, what is a hypotonic solution? A hypotonic solution will be a dilute solution. It will be a dilute solution or a solution whose water concentration is more than the liquid or the sap which is present in the cell, right? So whenever like this, when the cell gains water, when will a cell gain water? It will gain water when it is placed in a dilute solution. So this gain of water by the cell is called as endosmosis, whereas exosmosis is the withdrawal of water from the cell. Withdrawal of water from the cell is called as exosmosis, and this occurs when the cell or the tissue is placed in a concentrated solution. When you place it in a concentrated solution, then the cells are going to lose water. A simple demonstration of endosmosis and exosmosis is shown over here. Here what we do is, when you take raisins, what are raisins? Dry grapes, kishmish, it's called as kishmish in Hindi, right? So when you take raisin and you place it in a container that is filled up with water, what will you find? The raisins, which are dry grapes, will swell after some time. So how do they swell? It's because these raisins, when they are kept in water, raisins are dry. Water level in raisin is less when compared to the water which is there outside, and so water enters the raisins and that causes the raisins to swell, right? So this activity, the first one, shows endosmosis because water is gained by the system. Now in case of exosmosis, have a look over here. Exosmosis, when you take grapes, okay, when you take grapes, right, and you place the grapes in salt solution, leave it for some time. After some time, these grapes which are swollen, after some time, when you place it in salt solution, after some times these grapes are going to shrink. And why will they shrink? They will shrink because when you're placing the grapes, they have got higher water content per unit volume, per equal volume. When you take the grapes, have higher water content when compared to the solution which is there. The solution outside the grapes is having a lesser water content. Lesser water content means what? When you take equal volume of the sap which is present in the grapes and the solution which is in which the grapes are put, the water content or free water molecule outside is lesser when compared to the inside the grapes, and as a result of this, these grapes, they lose water to the outer medium. The grapes lose water to the outer medium, and as they lose water, these grapes, they shrink. Okay, let's now discuss another activity to demonstrate endosmosis and exosmosis. So, in this activity, we take an egg. Now, egg has got an outer shell. Just below the outer shell of the egg, there is a thin membrane present which holds the albumin and yolk inside. So, in this activity, the outer shell of the egg has to be removed. So, this outer shell, the white shell, hard shell has to be removed, and this is done by placing the egg in a beaker containing hydrochloric acid. So, we take an egg, place it in a beaker, and in the beaker we pour hydrochloric acid. When you leave the setup for some time, what you find is that the outer hard shell gets removed and the inner content does not spill because that is held by the outer thin membrane, which is this thin membrane. This behaves now like a semi permeable membrane. Okay, this will be behaving like a semi permeable membrane. So now what we do is we start our activity. So such kinds of de-shelled eggs with a thin membrane, two we have to make, and after this what we do is we take two solutions. Okay, one will be a dilute solution, or you can take just two containers. In one container we put water and immerse the egg in that, the egg which was de-shelled, and in the other beaker or container we take sugar solution. You take sugar solution, and in sugar solution you have to put the other egg. Now you have to wait for around 8 hours. So after 8 hours, when you have a look at the eggs, what you find is the egg that was placed in water. The egg that was placed in water, it swells. This egg swells, and it swells because it takes in water by endosmosis. Okay, so this takes in water by endosmosis. Look here, this first setup where the egg is placed in waters which will be serving as hypotonic solution, about which you will be learning. It's actually a solution whose water concentration is much higher than the inner content. So because of this water, egg being placed in this water, what happens is that water through that thinner membrane which holds the inner content enters by endosmosis, and as a result of this the egg swells. This egg, it swells. Next, the egg that is placed in the hypertonic solution, the egg that is placed in sugar solution, I told you that place another egg which whose outer cover has been removed by placing it in hydrochloric acid. So this egg which is placed in the hypertonic solution, it's going to lose water. Why is it going to lose water? Because sugar solution, it has got lesser water content when compared to the water in the egg, and because of this egg loses water by exosmosis. It loses water by exosmosis, and as a result of this the egg, over here you see the egg shrinks. Okay, so here is the egg that has shrunk because it's placed in a concentrated solution, and this shrinks because of exosmosis. The egg that is placed in water swells, and that is because of endosmosis taking place. So this activity clearly demonstrates endosmosis, that is entry of water into the medium, and exosmosis, that is loss or withdrawal of water due to exosmosis. Now study about the behavior of cells when they are placed in different kinds of osmotic solution. Now osmotic solutions are of three types. There are three types of osmotic solution, that is hypotonic solution, hypertonic solution, and isotonic solution. We'll see what happens to the plant cells and the animal cells when they are placed in these three types of osmotic solution. First, let us study about the hypotonic solution. A hypotonic solution is a solution whose water concentration is more than the cell sap. What does it mean? Let us see here. Let us take a just have a look at this diagrammatic representation. Here you have got a cell. This is a plant cell, and outside the cell, if there is a medium, there is a medium. Okay, now this medium that is there outside the plant cell, if it has got higher water concentration, water, like if you take equal volume of this outer solution and the cell sap, you try to recall, you have learned in class 8 that the fluid that is contained in the vacuole, that's called as a cell sap. So when you take equal volume of the two, if you find that the water concentration in the outer medium is higher when compared to the inner medium, in that case what will happen is that water is going, there's a difference, so water, there will be movement of water, and since there is a, the cell has got a plasma membrane, the cell has got a plasma membrane, and the solution outside and inside are different, what you find is that water molecules will start moving, water molecules will move inside, and also the cell is going to lose water. But since the water concentration outside is higher when compared to inside, inside the water concentration is less, what you find is more of water is going to enter the cell when compared to the water that's leaving. And so the net result of this is going, what is going to happen is that the net result will be that the cell gains water, the cell gains water by end osmosis. There is a gain of water by the cell. Okay, and as a result the cell swells. So here what happens in case of hypotonic solution is that in hypotonic solution, when a cell is placed in a hypotonic solution, that is around the cell, if the medium which is there, the medium can be the sap of the cells which are adjacent to these cells, it can be that also. So if the water content outside is higher when compared to inside, the cell gains water, and as it gains water by end osmosis, and when it gains water by end osmosis, the cell swells, the cell will swell, and in this condition the cell is said to be as turgid, it is said to be as turgid, right? So this happens. Now a plant cell, when you see, like both, like here this picture shows that the condition of animal cell and plant cell, right? Now in case of a plant cell, the plant, like when both these cells are present in hypotonic medium, both the cells are going to gain water. But what the difference that you find is that as animal cell has got its outermost covering, these are the RBCs or the animal cells. So as animal cells have got outermost covering of plasma membrane, as they go on gaining water due to end osmosis, these animal cells are going to burst. But this does not happen in case of plant cells. The plant cells will not burst. Okay, the plant cells do not burst when placed in a hypotonic medium, and that is because the plant cells have a cell wall. So the cell wall prevents bursting of the cell. So look here, the reason why the plant cells, not just the plant cell, but the plant cells, bacterial cells and fungal cells will not burst when they are placed in a hypotonic solution, even when there is end osmosis occurring, is because these cells have got cell wall in them. Okay, and the cell wall prevents the bursting. How? What? Let us see the reason. When placed in hypotonic solution, water will enter the cell. This is already told to you, water will enter by end osmosis, and when water enters by end osmosis, the protoplasm inside swells. Here what you find is that when water enters by end osmosis, the protoplasm, you know what is protoplasm, the inner content of the cell is called as a protoplasm. So this protoplasm which is present inside starts swelling, and when the protoplasm swells, what happens is the protoplasm exerts pressure on the outer wall. The pressure that is exerted by the swelling protoplasm is called as turgor pressure. But despite of the turgor pressure, the cell does not burst, because at the same time when the swelling protoplasm is exerting pressure on the wall also exerts an equal and opposite pressure on the swelling protoplasm. So as protoplasm exerts pressure, when protoplasm exerts pressure, the wall also exerts an equal and opposite pressure, and because of this pressure exerted by the wall, the cell does not burst, and end osmosis stops after some time. So after some time, this end osmosis, which is told to you, so entry of water or end osmosis will stop in case of a plant cell. So look here, water, I'm just repeating it again, the water enters by end osmosis when the cells are placed in a hypotonic solution. The protoplasm inside swells and exerts pressure on the wall. This pressure that is exerted is called as a turgor pressure. The wall in turn also exerts pressure on the swelling protoplasm. As a result, end osmosis stops after some time, and this prevents the cell from bursting. How what happens in an isotonic condition. First, an isotonic solution is a solution whose water concentration is same as the cell sap. That means that if you take a condition of this sort where the solution outside and the sap inside has same water concentration, so because of the presence of the plasma membrane, which is a semi-permeable membrane, this will allow osmosis to occur. Means what? Water from outside will enter the cell by end osmosis, water from inside is going to go out. But since the water concentration outside and inside is same, the number of water molecules entering the cell is equal to the number of water molecules leaving the cell. And so, as the number of water molecules entering the cell is same as the number of water molecules leaving the cell, the net result is that there is no net movement. There is no net movement of water molecules, and so there is no change in the size of the cell. There is no change in the cell size of the cell will not change. So this happens in case of isotonic solution. Now let us study about the hypertonic solution. A hypertonic solution is a solution whose water concentration is lesser than the cell sap. Means what? Just see this diagram. If suppose you have a plant cell in a medium, and this medium has got lesser water concentration when compared to the concentration of sap. So water content in the sap, the water content in the sap is higher when compared to the medium which is there outside, then this medium will be said as hypotonic solution. In this case, as the constant there is plasma membrane, and the systems are separated by plasma membrane, you find water is going to enter and leave. Okay, but then since the water concentration outside is less, there is less water concentration outside when compared to more water inside, and already I have said you, whenever there is a difference, water will move from higher water concentration to lower water concentration. So the what you find is that the net result is that the cell is going to lose water, and the cell loses water by exosmosis. So the net result is that cell is going to lose water by exosmosis. Here what happens is that the cell gains water and loses water, but since outside the water content is less and inside the water content or concentration is more, the number of water molecules leaving the cell is quite high when compared to the number of water molecules entering the cell, and so the net result is that the cell loses water. The cell is going to lose water by exosmosis. The cell loses water by exosmosis, and so the cell starts shrinking. The cell will shrink. An animal cell is going to shrink completely, but since the plant cell has got an outer rigid wall. The plant cell has an outer rigid wall. There is a cell wall also present, and cell wall is rigid. It's not going to shrink much. So after you leave this cell in this medium for certain period of time, the cell wall will not shrink, but the protoplasm will start shrinking, and it is going to move away from the cell wall. And in this condition, the cell is said to be as plasmolized. Look here, in this condition, when water has lost to the outer medium, which has got a low water concentration, the protoplasm shrinks, moves away from the cell wall. The protoplasm moves away from the cell wall. Cell wall is not going to shrink much, and so in this condition, when the protoplasm has shrunk due to excessive exosmosis, the cell is said to be as plasmolized. Okay, the cell gets plasmolized, and this phenomena is called as plasmolysis. So plasmolysis can be defined as shrinking of protoplasm. Look at this definition. It is the shrinking of protoplasm due to exosmosis when the plant cell is placed in a hypertonic condition. Okay, so when a plant cell is placed in a hypertonic condition, this happens. Just look at this picture. This shows the different stages of plasmolysis. First, what is there is that the cell, when this is a normal cell, now when there is hypertonic medium, that is a medium with lesser water content outside when compared to inside, then the cell starts losing water, and as it loses water in different stages, you can see in this stage, in this particular stage, the protoplasm has moved away, then more shrinking occurs in this stage, and finally the protoplasm will totally withdraw itself from the cell wall and shrink to one side, and in that case the cell is said to be as plasmolized. So plasmolysis is shrinking of protoplasm due to exosmosis when the cell or the plant tissue is placed in a hypertonic medium. It is placed in a hypertonic medium, right? So this is what is called as plasmolysis. Now a plasmolysed cell, when the cell has got plasmolized, you can deplasmolize the cell. That is, you can reverse the process. This process can be reversed back. How? When this particular cell, you place it in a hypotonic medium. So that means you just, the tissue that you have which has got plasmolysed cell, if you place it in dilute solution, then the reverse process will occur. The cell will then gain water, and after some time it will get deplasmolized. So a plasmolysed cell can be deplasmolized when it is placed back in a hypotonic solution. Here is an activity. You see here, look at this activity once, where later on in the lab also you will be performing this. You will be viewing the plant cells, onion peel cells. So these are the, like if you take a peel of onion and view it under microscope, when you take a drop of water you place on the onion peel and then the slide you place under microscope, you will find the cells in this way, right? The cells are all turgid, they are swollen because they have been, the tissue has been placed in a drop of water, and that water will behave like a hypotonic medium. Now if you take out that water and in the same slide, in the same tissue, over the same tissue, if you pour sugar solution, okay, and just wait for some time, and then again you view it under microscope. When you view it under microscope, you will find you will be seeing the cells in this way. And what is happening over here is that you can see this protoplasm in the cell has withdrawn away from the cell wall, and so the cells over here have got plasmolized. These cells have got plasmolized. This is an activity to demonstrate plasmolysis. Let us now differentiate between endosmosis and exosmosis. So look at the difference between endosmosis and exosmosis. Endosmosis is the osmotic entry of water into the cell when it is placed in a hypotonic condition, whereas exosmosis is the osmotic withdrawal of water when the cell or the plant tissue is placed in a hypertonic solution. Second difference is that when the cells are placed in a hypotonic solution, endosmosis will occur, and as a result of endosmosis, the cell swells. The plant cell will swell but will not burst because of the cell wall, but animal cell is going to burst because of endosmosis. Whereas in case of exosmosis, this will happen when the cell is placed in hypertonic solution. So in case of exosmosis, the plant cell will get plasmolized. The plant cell is going to get plasmolized. Okay, and the animal cell will lose water and will shrink. So this happens, and this occurs because of loss of water, that is due to exosmosis. So these are the two differences between endosmosis and. Now let us quickly differentiate between the three types of osmotic solutions, that is hypotonic, hypertonic and isotonic. Now just a recall of what all you have learned so far. Hypotonic solution is a solution whose concentration, water concentration, that is concentration of water when compared to the cell sap. So when compared to the cell sap, the solutions whose water concentration is high, that is called as a hypotonic solution. Whereas a solution whose water concentration is lesser when compared to that of the cell sap, this solution is said to be as a hypertonic solution. Okay, whereas an isotonic solution, the isotonic solution is a solution whose water concentration is same as the cell sap. There is a spelling mistake over here. This is hypertonic. Okay, so a solution whose water concentration is same as the cell sap is said to be as an isotonic solution. Now let us see the next difference. When a cell is placed in a hypotonic solution, then the water enters the cell by endosmosis. But when a cell is placed in a hypertonic solution, it loses water by exosmosis. And when a cell or a plant tissue is placed or animal tissue is placed in an isotonic solution, there is no net movement of water molecules. The third difference is as a result of endosmosis. When a cell is placed in a hypotonic solution, the animal cell will burst, whereas plant cell will swell and become turgid. Okay, both the cells will swell. Animal cell is going to burst, whereas plant cell will become turgid. It will swell and become turgid, but will not burst because of the presence of cell wall. Right. Now, in case of hypertonic solution, when a plant cell or a tissue is placed in a hypertonic solution, when it is placed in a hypertonic solution, what you find is that the animal cell is going to shrink. The animal cell will shrink, whereas plant cell will get plasmolized. The plant cell is going to get plasmolized. This is very clearly seen in this picture. It's quite clearly seen in this picture. This one, in hypertonic solution, the animal cell is going to shrink, whereas plant cell is going to get plasmolized. Now, in case of isotonic solution, since there is no net movement of water molecules, there is no change in this shape or the size of the cell. Okay, there is no net movement of water molecules, so the size of the cell will remain the same. Okay, so here these are the three differences between the three types of solutions. The hypotonic, you find plant cells swelling, animal cells it sliced or it bursts. In isotonic solution, the cells remain normal, and in case of hypertonic solution, animal cells shrink, whereas plant cells will get plasmolized. This is because of exosmosis. Endosmosis occurs in a hypotonic solution. No net movement of number of water molecules entering and leaving is the same in case of isotonic solution, and there is exosmosis taking place in a hypertonic solution. Now, let us learn about another type of transportation that is called as bulk transportation. In huge amount also materials can enter into the cell. I have so far discussed about the passive transportation, which is the movement of molecules of solid, liquid and gases. along the concentration gradient, that is where the cell does not spend energy. Then in brief I had discussed about active transportation, that is when the molecules of solid, liquid and gases moves from the region of their lower to the region of their higher concentration. In that case the cell has to spend energy. And then there is bulk transportation, that is in larger amount also the cell can take in materials. And here this one of the way of getting in material in bulk is endocytosis. So let's see what is endocytosis. Endocytosis is the process of cellular ingestion. It is the process by which the cell takes in food by making a cup shape invagination. Here you see, here is the definition of endocytosis, where you find it is a process of cellular ingestion by which this plasma membrane folds inwards. The plasma membrane folds inwards and brings the substances into the cell. Okay, so this is what is called as endocytosis. This topic I have already dealt in the previous video also. Amoeba feeds by endocytosis. So when the food particle comes closer to amoeba, the plasma membrane folds. It produces pseudopodia. Okay, and when it produces pseudopodia, a cup like structure is formed. You can see here there is a cup shaped structure formed. An invagination is produced. This is because of the pseudopodia that are produced by plasma by the cell, and the pseudopodia will totally encircle the food. You can see here the pseudopodia totally encircling the food, and thus this results in formation of a food vacuole. Okay, where the food is digested. So food vacuole is formed, and inside this food vacuole the food will be digested. So this type of cell feeding, where the cells take in food by making a cup shape invagination, and that is because of the pseudopodia being produced, this is what is called as endocytosis. So that's about how transportation occurs in across the plasma membrane. Now here's an assignment. On the basis of the video that you have seen, there are some questions that are to be done in your class notes. These questions are: the first question is differentiate between osmosis and diffusion. The next question is what is osmosis, and differentiate between endosmosis and exosmosis. Third is explain diffusion with the help of an example. Next is why doesn't a bacterial cell burst when it is placed in a hypotonic solution. And last, it is differentiate between hypotonic, hypertonic and isotonic solution. Thank you for watching the video.